then there is either the structural difference in the chromosome or either the numerical difference in the chromosome. We find these disorders, either the structural difference in the chromosome or either the numerical structural as well as numerical are structural as well as numerical differences which cause these chromosomal abnormalities. It could be extra and numerical causes when there is an extra chromosome over there or one less chromosome. When you say it is 23 pairs, it could be an extra chromosome or and one removed from that. When you say the structural differences, the structure of the chromosome may be like duplication, deletion, because of there might be short or long additions, this kind of things add upon the structure. So basically in maternal serum markers, we would be looking into these disorders: trisomy 21, trisomy 18, trisomy 13, TS9 non-hydrops, that is nothing but Turner syndrome, and preeclampsia. So what is this trisomy? Anyone is aware what is trisomy means?
Ma'am, additional chromosome.
Exactly. So tri states that it's 3.
Unpaired chromosome.
3.
Okay.
Tri is 3.
Tri is 3.
As you say, double is 2, tri is 3. So instead of you see a 2 chromosome at that particular, that is at 21 chromosome, there should be 2. Instead of that, you have another chromosome that makes triple. That is trisomy 21. That is 21 place. I mean like 21 pair. You said there is 23 pairs, right? So there should be at the 21 chromosome, there should be 2 chromosomes. That is a pair of 21 chromosome, where another chromosome is added to that. That becomes trisomy 21, which is nothing but a Down syndrome. Yes? So there it becomes 47 chromosomes because of one extra added chromosome. So in trisomy 21, the features like phenotypically, babies would be after birth. Babies would be with intellectual disability, flat face. I mean, you can recognize them by looking at their face. They will have high set eyes, flat face, and they'll have intellectual disability, and to some extent there would be even cardiac related issues also. Okay. So in the same way, trisomy 18 says about the chromosome 18, 18th pair chromosome has extra chromosome. Nothing, it is called as Edwards syndrome. The disorder name, the syndrome name is Edwards. All these disorders are named by the scientists who has discovered it. So trisomy 18, where the 18th pair of chromosome has another extra chromosome, which again makes into 47 chromosomes. In the same way, trisomy 13th chromosome having an extra chromosome, and this is called as Pathankot syndrome. Okay, preeclampsia we'll discuss little later. Okay, these are the disorders covered in maternal marker serum. According to, yes, trisomy 21, whereas the Down syndrome, it has I mean to say that after birth baby can live. There may be some intellectual disabilities and heart defects, abnormal heart conditions, but they can lead a normal life. Whereas coming to trisomy 18 and trisomy 13, there is no chance. Maybe a year, 2 years or so. They have very low birth weight, failure to thrive, small head, severe heart defects. And in Edwards syndrome, you can see even the cleft lips. There will be a cleft in the lip. or even spina bifida, which is nothing but a neural tube defect. And you are very much aware that neural tube defect, anyway you find a neural tube defect, like it is like there's very high mortality rate in the neural tube defects. And coming to trisomy 13, there would be almost all organs are affected. Say your kidney. Sorry, I didn't get you properly. Yes, you are telling, you have the answer. You are saying it is genetical. So when the genetical disorders occur, they could be two. It could be genetical, or hope you remember cell division. Everybody remember cell, right? So there is a process where in the metaphase, meiosis, there is this division where the spindle fibers pull backwards the chromosomes when there is a division of the cell. At that particular moment, if this chromosome is not moved towards the poles as it has to move, the pair completely moves towards the one side, then there is a chance of that particular cell having extra chromosome. Is that fine? Yes. So indications of the test just now, like Savinder was asking, like why? If the maternal age is above 35, then there would be a chances of Down syndrome, or else previous child with a chromosomal abnormality, or if it is twin or multiple pregnancies. As I said, at the point of cell division, there are more chances. If it is a twin or multiple, at the time of cell division, this may happen. So there is more chance. It may or may not. If it happens, then consecutively, like this disorders would be out. If there is a family history of single gene disorders, this is again genetical. So it is like already known. It's running through the family. Or family history of neural tube defects, or women with a bad uh, obstetric history may also have this risk. So all these, whenever you see this in the patient, like twins or maternal age is high or any chromosomal abnormality with the previous child, then doctor will recommend for maternal serum markers. Or it could be a consanguinity also. So what all we check in maternal serum markers? Anybody have any idea, like what are the parameters we check? You said
Yeah,
like it's double, triple, and quadruple.
in double marker, it is Papa and 3-beta HCG.
Okay.
And in triple
You
marker,
can
it
see on the
is,
screen. Yes, you can see on the screen.
yeah, okay, yeah, it is there. Okay.
So
Triple, it is
what
ASC,
is that anybody
HCG.
aware that what are this? You are telling it's PAPE, it's P beta HCG, AFP, estrol. What are these?
So, ma'am, 3 beta HCG is like human chronic gonadotropin hormone.
Mm-hmm.
This is produced by placenta,
Mm-hmm.
and this is normally used to detect Down syndrome.
Mm-hmm.
And PAPPA is nothing but pregnancy associated plasma protein
Mm-hmm.
that will again Down syndrome trisomy 21 they will counter.
Exactly. So almost the screen says everything. You guys can speak it out. Anyway, so I just wanted to say that these are nothing but, as we were talking in the last session, like I was talking about organic compounds, metabolites. So in the same way, these are nothing but the hormones and proteins specially produced during the pregnancy. Okay, based on the levels, the rearrangement of these hormones and proteins, we can say, or we can conclude, or we can interpret the diagnosis, whether it's 21, 13, 18, or preeclampsia. Got it? So, like double, triple, quadruple, pentamarkers, and first trimester quadruple. So, these are the analytes. These are the hormones or the proteins which we look into particular trimesters, first trimester and second trimester. So double marker has free beta HCG and pathway, nothing but a protein and a hormone again. Triple marker AFP, HCG and UE3 estrol.
Is the elevation or the deficiency causes these anomalies?
It depends on the elevation as well as the low level. See, free beta HCG when you take, it increases.
Okay.
Okay. Okay. When you take estrol, it decreases.
Okay. Okay.
Right. It has to increase. Basically, it has to increase. If there is a disorder, they would be low. So
Okay.
it will not show to the optimum. I'm just giving you rough value. So if it has to be 20 micromoles, say suppose 20 IU. This is not correct figure. I'm just, for example, I'm explaining 20 IU at a week of 14. Okay. So,
Okay.
but if there is a chromosomal abnormality in the baby, right, then instead of having 20, it could be only 9 or 10.
Okay.
Right. So only with this, only with these hormones or proteins which we are detecting through an specialized equipment is not sufficient for the diagnosis. So that's the reason I have given in this screen. Just look into the screen, demographic and sonographic details. So what are these demographic and sonographic details? Can anybody say? Yes. Hmm. Hmm. Right. So NT, NB, and CRL, BPD, right? We keep asking for this details. Once we receive the sample, our technician keeps mailing you saying that we need CRL value, we need BPD value to calculate the risk, we need NT value, or we need NB, right? So these demographic and sonographic details along with the serum marker values. Serum markers are nothing but these ones, PAPA, free beta, HCG, all these are serum markers. So along with that, only detected in, only can suggest into which risk, like it's 21 or 18 or 13, whatever it is. Sorry guys, I've missed. See, we are using software, I mean, like instrument is Victor 2D, which works on fluorometry, immunofluorometry. These are all our immunofluorometry assays, and the software which we are using is Life Cycle. It's a higher version of, we are using version 7 of Life Cycle software. Please note it down, guys. I somehow missed adding it into the slides. We are using Life Cycle software version 7.
Life
Right.
cycle.
Yes. So we have in the market, we have Prisca as well as Life Cycle. Life Cycle is from Perkinelmo. Victor 2D or Autodelphia, these instruments are from Perkinelmer. It's a closed system. We use life cycle software for calculating the risk. So basically, instrument is used to note down the readings, like what is a concentration of this particular proteins or hormones present in the serum sample, then this results are transferred to the life cycle software where it calculates the results. In this life cycle software we have to give all this demographic and sonographic details, where it calculates with the help of demographic, sonographic and serum markers and gives final risk. Either it could be normal or it could be diseased.
Ma'am,
Okay.
what is
Sorry.
81%?
Sensitivity. Savindra, there's some disturbance in your voice. You're asking about the sensitivity of the test? Hmm. It is almost 90 above, 90% above the specificity and sensitivity goes. Okay. Anyways, this is screening. So we can't give 100% to the any screening tests, the sensitivity or the specificity.
Hello.
Yes. Yes.
Hello.
Yes, Kunal.
Yeah, I have question. As you said that we are using Vector 2D machine, and that is one another Autodelphia, as you said,
We are
right?
using only vector 2D.
Uh,
Autodelphia we are not using.
yeah, that is my question is, both are from Perkin Elmer,
Yes.
as you said.
Yes.
So there are many local lab who are using the Autodelphia. So is there
Yes.
any, the result changes, or is there any, price issue?
There
Because
there
as
would
I
be more
heard,
higher version. Autodelphia is a higher version, so obviously the sensitivity of the technique would be more. The instrument would be more than vector 2D.
Autodelphia is, higher version of Vector 2D, right? As you
Yes.
said.
Yes. Yes.
Okay. Okay. Sure. Thanks.
So so which weeks are preferred for the double marker, triple marker, penta, or the second trimester? You can just have a look over here. So gestational age 10 to 13.6 weeks is preferable for double, first quad, penta, and preeclampsia. 10 weeks to 13.6 weeks. And the second trimester 14 to 22 weeks. Second trimester includes triple marker and quadruple marker. You can just look into the analytes, I mean like hormones or the proteins which we analyze in which test. Double marker, that is PAPE, free beta HCG and NT. NT is must for calculating the risk. So that's the reason I have specified like demographic and sonographic results are more important to calculate the risk. Just have a look at this. You guys can note it down because there would be questionnaire based on this.
Ma'am, in this slide, second trimester screening
Mm-hmm.
uh, for triple and quadruple marker, it is recommended that we can do this test after 20 weeks, as it is mentioned 14 to 22 weeks, because as per the new guidelines, the government person means they can abort the sample after not after 22 weeks, right?
Yes. Is that clear with the slide? Yes. Hmm. 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 Scan. Okay. Hmm. 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 I'll tell you, Savinder. This must be because the scan report must be stating the different week. See, when you look into the scan report, they would be stating according to LMP, according to CRL, according to other some sonographic based observations. They will say into which week it is falling. So, if you look into the CRL based gestational age, then that will obviously fit into the criteria. LMP you should never check because it is like it may differ two days to 1 week difference may be there with the LMP. So you may clear that into which gestational age you have looked. Based on that, you have to collect the sample. If the CR and one more thing, if the CRL value is also low, it is not fitting. Then obviously we prefer to collect sample after a week, that is in the 11th week, to just avoid false positive results. Okay, can I move this slide because be clear with
Ma'am,
the
one more
slide in
question,
which
ma'am. Ma'am, one more question, ma'am.
yes,
Ma'am, in 14th week, can we collect the sample? Because I was aware that in 14th week we cannot collect any sample.
14 to 13 to 14. Sorry, yes. Actually, these people, like for from Perkinelber and these people, they have started saying that 14 to 22 weeks, 22.6, they can based on the moms. But you're true, 14th to 15th week, ideally we shouldn't collect any sample because there would be, see, in the second trimester, we are looking for AFP. If you observe in the second trimester, both the tests include AFP. AFP will have huge change in 14th to 15th week. So, preferably, we shouldn't collect samples in 14th to 15th week. But in case if there is any recommendation from the doctor or it has to be done, then you can collect and please mention. I mean, like anyways, we have the demographic results which you send. We can have over there. In case if there is a positive report, then obviously we would be asking for a repeat sample after 1 or 2 weeks, so to confirm the results. AFP would be there would be. Hmm, yes, exactly. uh, Maintaining the temperature we can prefer for 24 hours, but as the doctor said, he's true, after collecting beta hcg value changes for every hour. Got my point? Every hour there's a difference in the beta hcg levels, but we can say it could be 24 hours if proper maintenance of the condition, sample conditions, like it should be in 4 degrees. At least that will help in hindering the change. For lower amount of hindrance, I mean, like higher amount of hindrance is seen, like if you see that only 10 um, IU of beta hcg will be seen in the fresh sample, right? If it is there is a delay, then you could see 30 for 2 hours. In RT you could see 30, or after 48 hours you could see even 100 also. These value, don't take this value serious, I'm just giving example how would it increase. If you put it in a 4 degrees, at least that could show to somewhere around 50 or 40 less. So that's the reason we say proper transportation conditions are required, and free beta hcg, yes, there is change even for each hour also. That's the reason we consider the demographic results also. if all the demographic results are normal and only beta HCG value is high, then we can say that it may be because of transport or storage conditions. This beta HCG is high. Then we can take either repeat sample or else talk to the doctor, convey this. If he also is convinced with that, then we can say we can release a normal report. Got it? Shall we move on to next slide? Hope you have noted down this details. So what is the clinical utility? Why we have to go for maternal serum markers? We have already discussed to check the chromosomal abnormalities or the neural tube defects, or if there is any cardiac defects. So cardiac is like later. You can see it with the ultrasonic, like when the scanning is done. Okay, so these would be the utilities of the maternal serum markers. And what are the limitations? So first and foremost limitation would be it is a screening test. We can't judge 100%ly with the screening. As you guys remember, Saturday I was telling I can judge with this technique organic acids, amino acids, and even TMS also I can judge, because the instrument and the methodology which we are using may give you 100% results. I can say there's false positive of not 1%. But here, as it is a prenatal, we are taking the mother's sample. There should be enough of these placental hormones and proteins in that sample. It's just a screening, and you need the proper demographic results. If without demographic results, you can't judge into which disorder it would be falling. So that is the highest limitation for this test. So as with all, it is a screening test. We cannot judge based on only the serum markers. Okay. Then as it is a screening test and confirmatory is mandate for that, we need to go for either like NIPT or amniocentesis for confirmations. So workflow, this is as same. Like sample transportation, receiving in the frienders, then the sample is registered in the frienders and then sent to the lab. In the lab again we receive the sample, we give separate unique test ID for the sample, then we process the sample processing here involves, one second. Yes, sample processing here involves, it's a immunoassay, immunofluorometry assay, where for each parameter which you were looking, there should be a run again. For free beta HCG, you need to take a plate, add everything, process it. So for each parameter, there is each step. Each plate would be taken, 96 well plate would be taken. You add the sample into it, you add proper reagents into it, incubate it, wash, then add, just 1 second. You can see my screen, right?
Yes, ma'am.
Yes, this is the process which is followed for each hormone and each protein. This is just example of one. So there would be a solid phase where the streptavidin is coated onto it. To that we add biotin labeled anti. Here I have given PIGF. It would be everything like HCG for HCG, UA3 for UA3, PAPA for PAPA. Those are nothing but labeled antibodies. Okay. Then you incubate it. Afterwards, incubation period goes on for 1 or 2 hours depending upon the reactions. Afterwards, you just add UE labeled antibody again. Europeum. UE is nothing but Europeum labeled antibody. It is a sandwich model. So antibody, then you analyze, then the antibody, you incubate it. Then afterwards you add enhancer which removes the UE. So how much ever UE is removed from this reaction, that is being measured. So this amount of UE represents it in your report in the IU manner, or whatever the units it is, in the concentration manner. This UE value is measured.
Hello, ma'am.
This slide was done.
No, ma'am, no.
Uh, uh, I was showing you the other picture also. Was that clear?
Yeah, ma'am, that's clear.
Okay. So then that is the sample processing. Then after sample processing, then the samples are loaded into the 2D vector. Once we get the results from the vector 2D, then these,
Ma'am, previous slide is not explained, ma'am. Previous slide.
this is what I was explaining.
Next, okay, okay. The sample collection type, ma'am. The
No,
sample to report.
that is the next is
Okay, okay.
sample type is next. So hope we have finished till the dispatch. Everybody was there. I mean, like my team was there. We'll move on to next slide then. So sample type, you all are aware of sample type and that also. So may I know, like anybody knows that what sample should then be collected for markers? You are aware that which sample has to go, but which sample is not preferred. Anybody?
Amniotic, ma'am.
I'm sorry. I'm talking about serum. Which tubes are not preferred? I'm telling you, red and yellow tubes are preferred, but which are not preferred?
EDTA, I think.
Heparin,
EDTA.
EDTA, heparin,
Yes, EDTA, heparin,
heparin,
and citrate, especially citrate also.
citrate also, right?
Because citrate and EDTA are the chelating agents which hinders the binding. Just now I've shown you in a picture where the antibodies bind red colored tubes are preferred for serum collection. Is that clear?
Yes.
Yes. And sample should be transported at 2 to 8 degrees within 24 hours. 24 hours we have already talked because there would be change in the hormones. So target audience would be like, now we have received samples from Ankura, Base Hospital, MH, Jui Fertility, Continental Hospital. Okay, so you need to target the gynecologists and fetal medicine experts for maternal serum markers. In market we have you guys are aware, like who else, rather you are the best people to tell me who are in the market. Yes guys.
Lupin,
Yes. So what are the softwares used over there? Any idea?
Lupin doing dual marker in just 4 to 500 rupees.
Okay. What is
Apollo
the
also
software?
does
What is the
double
instrument?
master. Apollo also does the double master at 500,
Praska.
and Perkin Almer they are using, and they are also using Life Cycle 7 software.
Seven software. This is about LifeCell,
Apollo Diagnostics.
Apollo Diagnostics. Yes. So there are huge players in the market. So USP is like we have self-established
Yes, sir.
moms. Moms are nothing but multiples of median. Was talking about all the hormones and proteins we detect through this technique, Victor 2D. So is that we are giving only the value which we have got? No. We would be comparing it with the data which we have from all these years. So based on our data, we have lab derived moms. Okay, so we compare with this so that they would be less of false positives or false negatives. So this is how this we can say this is how we are prior in that, like it is one of our USP. And life cycle software 7, this is FMF approved. Okay, what is the cost? I didn't get. You're asking about the cost.
Cost is all, he has asked about cost of that machine. Money.
Sorry.
He's asking about the what is the cost of Victor 2D machine. How much we need to pay to buy that Victor 2D machine.
What is the use of the cost? May I know the reason? It may not. Only the instrument cost doesn't depend on all the pricing issues. CRP is based on all the we consider everything and then we give the costs. It is not only based on the instrument. Okay. Anyways, so we will report even twins and triplets also, but only if we have these details. This is done in double marker testing based on CRL, NT, NB, and biochemical markers. But we report separately for each fetus. Twin is 2 fetus, right? So we report twin 1, I mean fetus 1, one risk, and fetus 2, another risk. I mean, like whatever we get, we report. Individual fetus risk is being given in the report. So this is how our report looks. Hope everybody have seen our report. The green one indicates the negative and the red one indicates the positive and gives a high alert. We give all the demographic, whatever the information we have got is seen in the second page. We enter everything into it because then only we can get, then only the risk calculation is possible. This is how this picture is taken from the right cycle, this graphs. So we have already added, we have changed our report format also, we have added interpretation into it. Once you look into the interpretation, you will, any layman can understand like what is what, at which mom level or at which what is a cutoff, when we can consider it to be intermediate, low or high. Everything is given in the interpretation.